So Petr received his PhD at University of Chemistry and Technology, Prague in 2008. Afterwards, he did his postdoc at the Scripps Research Institute, La Jolla, USA, in 2008 and 2009, in laboratory of Professor Finn. Since 2013, he has been a head of synthetic nanochemistry research group at Institute of Organic Chemistry and Biochemistry of the Czech Academy of Sciences in Prague. His current research interests include nanoparticle surface chemistry, their biointerfaces, and their applications in biomedicine and sensors. In particular, he focuses on chemistry of diamond surface and development of diamond-based quantum nanosensors, the topic of today's lecture. Petr published more than 80 peer-reviewed papers. So, Petr, the stage is yours.
Okay, Olga, thank you very much for the invitation. I'm happy to actually contribute to this nice series, and I appreciate actually that you are organizing this. So in my talk, I will focus on the molecular transducers, which are actually needed for bringing diamonds to the level of a selective quantum sensing. The talk will have like different parts, and I will try to introduce the material from different views. In first part, I will just discuss diamonds in general, why we are actually using them. In second, I will briefly introduce how we can prepare and create NV centers in diamonds. In next part, actually, I will focus on the main topic, which is the transducing of the sensing to the NV centers in diamonds. And in the last part, I will talk about the biological aspects of this sensing. It means how can we get particles into cells and which difficulties are we expecting during that. So first, I would like to thank to my team and to our also collaborators, which you see on the bottom of the slide. I hope I didn't forget about some of them, because I have a couple of collaborators which are like in dynamic kind of like collaboration doing the projects together with me. So thank to all. I'm actually happy to collaborate with you. My laboratory, as Olga said, is in Prague, which is basically heart of Europe. It's in the center of Europe, is the capital of Czech Republic. And the institute is just about 2 kilometers from this nice Prague center. So we are in an old part of Prague called Davice, and this is our institute from the front. And from the back, actually, there is now a new building, which you can see here on the bottom. And my labs are here because this is dedicated to the synthetic chemistry, which we mainly do. So yeah, that's just for introduction. So first, I mention maybe things which are for many of you trivial, but I don't know exactly who is attending the lecture. So I will try to bring it like in general to kind of normalize the knowledge about diamonds. So what actually brought me to the diamond? Because I thought about different types of nanoparticles. For example, upconversion particles when I started my like independent career, and I thought like, well, I was definitely interested in cell imaging and working in the biological material. So diamond attracted my attention and actually kept me later for like now for maybe more than 10 years. And the reason is that I found actually that it's really like material with a low toxicity. This is something quite unique. Many materials can release toxic ions. Indeed, carbon cannot. But that's not only this point, because other types of carbon, you know, can be in certain perspective toxic. Diamond can be also toxic because always the toxicity is just given by the overall dose which you are using. If you drink too much of water, you can be poisoned even with the water. So That's the same for diamond, but the fact is that diamond is really low toxic. But also what is interesting, it provides different types of fluorescence, which are provided by the different types of lattice defects. Today we will talk about the NV centers, but there are many more of them. There are actually hundreds of them. Only some of them are useful for like bioimaging, for example, SIV centers, but there are also others. And people are investigating it now quite like extensively, this field, what can be used from the available number of the defects. So NV centers means these are like nitrogen and vacancy centers. So it's like adjacent vacancy to a nitrogen atom, which is an impurity in the diamond lattice. So this center provides emission from the biological point of view, very importantly in the so-called near-infrared window. So you see the red is the emission profile of the NV center, which is mix of two states, NV0 and NV minus. Everything which we will discuss today is actually about NV minus, because that's the active, let's say, spin active form of the center. And it emits in this window, which you see, the logarithmic actually absorption scale here. We are in the minimum of the absorbance of water, but also of the blood dyes and other actually pigments which are present in cells and tissues. So it can be better, it can be shifted exactly like on the 800. We are with maximum at about 700. But still, you can see the dominant part of the spectrum is actually reaching this. So that's quite useful. And we can do like background-free imaging using this, because the autofluorescence of tissues is quite low in this. And we can also gate the fluorescence of diamond, because it's the lifetime is much longer than the classical fluorophores. If you think about the cell as a source of green fluorescence, which is like omnipresent, lifetime is between about, let's say, 1 nanosecond to about few nanoseconds. If we gate it at about 10 or 8, 12, like that, maybe 15, we will get a very pure signal, like without almost any background, which is caused by diamond. So that's advantage, which is also not available in many other types of fluorophores. If you look here on the graph, which was measured by Philip Reineck, so this is actually showing that many of the different dyes which are emitting in near infrared have kind of comparable spectral quality to diamond. They are photobleaching quite quickly. Alexa is a molecular dye. It's like, at the same radiation conditions, it's within 1 minute, it's basically over. The other things like carbon dots, although they are quite stable, you see within just a few minutes, they are like gone. If you look on the diamonds and nanorubies, only these two kinds of fluorophores of stable, and we can extend the curve very long, for hours or even days without any change. This is very advantageous for the biological imaging. So if you see the carbon dose is actually rising up, it means they are also changing, which is unwanted. So it's not an advantage. Okay, so let's go to the other slide. Now I'm talking about the electronic structure of this NV center. So I don't know how much familiar are you with these schemes. It's called the Jablonski diagram, and you see energetic levels of the excited state of the diamond and the basic state. So what we do, we basically irradiate by a laser this basic state and excite the electron by photons in excited states, and from these excited states we are relaxing. And there are two ways how can you relax. The first one is a radiative relaxation. So it's basically going this way. Back if we are relaxing to the same level, we are emitting the light which we can see as a fluorescence. On the other hand, there is the other way, which is shown here, and this is the singlet state, which is non-radiative. it's basically kind of thermal relaxation. If the electron is relaxing via this pathway, it's not producing any light, and it's actually kind of weakening of the yield of the fluorescence which we get from the particle. Okay, now even a bit more difficult. So the basic state is now split to two sub-levels based on the magnetic spin numbers. Is the number 0 and plus minus 1, which is degenerated. It means it has same energetic level. So if we look on detail here, so the energetic distance between this is actually very low. It's quite close, and it's not in the visible light. It's actually in the microwave spectrum. So if we irradiate this using the microwave, we can excite the electrons from the MS0 in the basic state to MS plus minus 1 in the still basic state. But now, the very interesting thing, if we have a look, the only allowed transitions are from 0 to 0 or from plus minus 1 to plus minus 1. So, and now you see the probabilities as is depicted by these arrows. They are very different for these spin states. From 0, actually the probability is quite low. So if we are exciting from this very basic state, the photon, like, it's not going this way. Sorry, the electron is not relaxing this way, and we got the highest possible yield. On the other hand, if we combine irradiation, like photon excitation from this plus minus level to this one, and we combine it together with the microwave irradiation, so we are exciting from this level to this level, and now we have a substantial amount of the electrons relaxing via this singlet state. And this is causing drop on the fluorescence. It looks like this. So if you have fluorescence, and this is a microwave scale, so if you are tuning the microwave frequency, once you will reach this actual resonance frequency between these two levels, there is a drop in the fluorescence because the electrons are energetically relaxing via this way. So that's what we see here. And actually, to make it even more complicated, if we put the diamond in a magnetic field, you can split these two levels, the plus minus 1, in so-called zero field splitting. And what is happening? Here we get like two another levels, which are now can be distinguished, and the distance between them can be also seen. So we have basically then like imprinted the magnetic response of the NV center in the microwave-based spectrum, but we read it using a fluorescence, that very unique actually setting, which is not available in the other materials. I spent some time with this because I see this. I think it's quite important actually to emphasize these all details, which we will talk later about them. Okay, so based on this, we have very unique and different features and possibilities which is provided, because the other like fluorescent molecules are not providing these options. So we can do magnetometry, we can watch the position, we can like sense the presence of paramagnetic species, even temperature, and we can do some smart kinds of background-free bioimaging, not only by the time gating, but also by other pulse methods. So all of this can be read optically, which is actually, for biologists, it's very important, because biologists is using typically with cells of confocal microscope or some live imaging for tissues. So the light is a, let's say, good way how to investigate biological samples. So we can do basically even single molecule like NMR or EPR. We can place the diamond in a scanning like AFM tip and investigate magnetic domains or structures, or we can work in the cells. So in the cells, if we place diamond there, we can sense different things. We can sense viscosity, rotation of the diamond. We can sense temperature, but also paramagnetic species. So, and what I am actually going to show you today, even the species which are not paramagnetic. The point is how to actually translate the language or the presence of the molecules which are not paramagnetic to read out using a paramagnetic NV centers, which is everything translated into the magnetically dependent fluorescence, right? As I was showing in the past slide. Okay, but first, if we want to actually use these particles, we need to create them. We need to create NV centers. So actually, nowadays, several groups are working on this worldwide, including us. So it's actually still there are many details how to improve the quality of the diamonds, how to improve the yields of NV centers, how to improve the yields of the particles. So for some projects which we thought we can basically do, we needed some larger amounts of diamond. So we thought how to create some big quantity of them. So the classical, let's say, or now well-established way how to create NV centers is to use diamonds containing nitrogen impurities, irradiate them using high energetic particles, and then you actually create vacancies. You physically remove the carbon atoms from their position in the lattice. This is not enough. You need later to heat up any of the particles, which is causing migration of the vacancies. And if they are able to find the position of nitrogen atom, they recombine in NV center, in the nitrogen vacancy center. And because within the range, in the temperature range which we are using between 600 and about 1000 degrees centigrade, these centers are thermodynamically stable. They don't move further in the particles, and they stop there. So that's the approach. You can use protons, electrons, alpha particles, like different approaches. Each has certain advantages and disadvantages for the creation. For example, electrons are penetrating deep in the matter, centimeters in depth, and also creating quite homogeneous NV centers. Originally, we were interested in protons, but later we were using also alpha particles. Here, there is a kind of interesting limitation, which is caused by the relatively low energy, although the energy is very high. So there is a penetration depth of the energetic particles into the mass. So you create some target where you put a layer of diamonds, which is typically just about 1 millimeter thick for protons, for example, or 2 millimeters, not more. And you irradiate by the beam of the accelerated particles. And what you see, here is like the depth of penetration, and here is normalized energy deposition. So this is called Bragg peak. It actually showing that the most of the energy is deposited within quite narrow region, which is inside the target. So if we make, for example, 2 millimeter thick layer of diamond, most of the energy is deposited within just few tenths of the millimeters here in this region, and the others are basically not irradiated or just like little bit, or not at all here. So we worked in some aqueous solutions of diamonds, and we improved the homogeneity of the irradiation, but still the yield was about hundreds of milligrams per one irradiation, which is quite costly. So we thought how to improve it. And we actually utilized the nuclear reaction. So this is the approach which we take from the therapy of cancer, actually, because there is used so-called boron neutron capture therapy. If you irradiate the boron nuclei 10B, this one type of nuclei by neutrons, which are just of appropriate energy, is called thermal neutrons. The neutrons absorbed, and immediately the activated nuclei is split to lithium-7 and the alpha particle. Both of these particles are able to create vacancies. And when we inspected actually the energies of these particles, it was exactly those energies which we need for creation of vacancies in the diamond lattice. Is quite ideal for that. So what we did, we simply developed a procedure how to make kind of boron oxide glass with nanodiamond kind of composite. And the diamonds were very well dispersed in this kind of glass. It was basically transparent. So we milled it and filled it in a kind of capsule, which we exposed in a nuclear reactor to the beam of neutrons. This type of irradiation is very homogeneous because the flux of neutrons in the nuclear reactor is about 1,000 times or 10,000 times stronger than in any accelerator which you can create photons or electrons. So the fluxes in nuclear reactors are very high. And if you have a look on the irradiation geometry, it's actually providing distribution within a few micrometers from the source of the radiation. But now what we have, the flux is all directional, is very important because the neutrons are actually penetrating through quite easily through all the mass, and they are generating the alpha particles and lithium particles in all the mass in all directions. So we don't have now the setting that we are actually irradiating and we have the Bragg peak. The Bragg peak is occurring from each point of the mass. So we get very homogeneous irradiated particles. Each alpha particle is traveling in the way it's meeting about few tenths of the nanodiamonds, or even it's valid for the lithium. So this is causing the homogeneity. You can see it travels few microns, and within these microns we have this number of particles. So we actually saw it works quite well. If we took just diamond and irradiated without boron oxide, it was not providing almost any fluorescence. But with the boron oxide, we immediately get a very, very strong fluorescence, which is well known from the other irradiations. And if we compare it, for example, with the alpha or proton irradiations, we got roughly the same spectra for the larger or smaller particles. So we will say like, well, this works exactly, actually, we would believe. What was actually advantageous from technical point of view, we did not improve the quality of the diamonds, but we improved the technique of the irradiation. We basically obtained the same results in the few minutes of irradiation compared to the many hours which you need at the accelerators, which was kind of shocking for us because even after longer time we saw degradation of the quality of the fluorescence. So the 3 minutes was the shortest time for the handling of the sample. I believe it can be even shorter, but it doesn't matter. So we irradiated within 1 hour or half hour, I don't remember, about 100 grams of diamonds. So it was very quick and very cheap. So the quality in terms of for example, zero phonon line weight was comparable to the other irradiations. Here we compare with electron irradiation, and also zero field splitting was the same. So we were like, well, it looks like we can produce a lot of diamonds. So this was kind of new approach for us, at least, how to create particles irradiated. It works also for other types like silicon carbide particles, where we produce the other types of fluorescent centers. So yeah, that's kind of new technique for this. Okay, so I wanted to show you this because from my perspective, it's kind of the discovery which is in my lab. So I thought I will present it here. But now we have the particles. So the key question today is how to use them for quantum sensing, right? How to detect the species which are paramagnetic or they are also not paramagnetic. So for this, we need to design and construct molecular transducers. Back to what I showed you, actually, I did not mention one of the key technique which is used, and this is measuring so-called T1 time. Maybe you heard about this, what is T1 or T2 time. So T1 time is not related to this, the basic and excited triplet excitation. It's actually all related to the triplet state, which is basic. So we are talking here about this. Relaxation time T1 tells you how quickly the electron is relaxing from the level with magnetic spin number ±1 to magnetic spin number 0. So it's about this. And what is the feature of T1 that is very sensitive to the presence of the magnetic field? If there is any spin around diamond, which is in the proximity, and can interact in a magnetic way in spin interaction with NV center, is actually accelerating the relaxation from this. So it's shortening the T1 time. And that's the principle which is called like T1 relaxometry, or you can find it like in about this type of naming of this. So the other techniques, for example, splitting of this can be called as the optically detected magnetic resonance, or ODMR. It's also often seen in papers. So there are different approaches how to utilize, again, this whole spin system. And now we will talk about the T1 relaxation, so in the rate of the from ±1 to 0. Okay, so here are few examples. There are actually many more papers on this indeed. Like people are really investigating it now extensively. If there is a like paramagnetic species, for example, here, like gadolinium nearby NV center, or here is manganese, like manganese 2+, which is paramagnetic. There are other gadolinium, or here are ferritin, which contains a lot, about few thousand of the iron 3+ ions. All of them you can detect based on the like changed T1 time if they are in proximity of NV center. If you look in ensemble sense, this was actually a single NV detection. If you look at ensemble NV, again, you can detect, like, for example, gadolinium, which is have the highest spin actually from all the ions which you can find. It's 7.5. It's really high. So you can do ferritin imaging and so. So these approaches are very interesting and demonstrated how sensitive is actually NV center, like how sensitively it can feel the presence of paramagnetic species. The problem of these measurements is that we cannot distinguish them. We cannot tell if the change in the T1 is caused by manganese, gadolinium, ferritin, or other paramagnetics. So this is not distinguishable. We just can measure a specific quantity by optical means. I'm now talking about the T1, right? So you can measure either T1 or T2, and you can distinguish. So like several teams, I just exemplified by my colleague Cheng Li team and Renbao Liu from Hong Kong. So they came up with a interesting like idea to create on a diamond a stimuli responsive polymer, and to attach to this polymer magnetic nanoparticle, like super paramagnetic particle. So this is responsively shrinking or swelling upon a stimuli. So you are changing the distance between the NV centers and the particle. So based on this, there is the expected change of the like ODMR spectrum or T1 relaxation. And indeed, actually, they observed this very nicely. This is ODMR spectrum. You saw it already previously as the dependence of the normalized fluorescence in this way on the frequency of the microwave, field which you are applying. So it's the transition between the ±1 and 0 in the triplet basic state. So what they saw, they have thermoresponsive polymer which is shrinking upon the heating up. So what they saw, upon increase of temperature, they saw nice actually change in this difference in these frequencies. So the spectrum was broadening. Is indeed broadening because you are actually approaching magnetic field towards the NV centers. So this was kind of like a molecular actuator which was mediating measuring of one quantity which can be also measured by NV center directly, but it was translating it to the language of molecules. So we have a basically chemical event which is occurring nearby the diamond, and we can have it like optical readout of this. So we also addressed this, but we were not measuring temperature, but we looked on measuring on pH and redox potential. So what we designed was different kind of polymer, which was not thermoresponsive, but was designed to have a cleavable linkers which can release paramagnetic ions. As you saw on most of the previous cases, people are using gadolinium. This is the element of choice because it's really have strong interaction with the NV center thanks to its strong magnetic moment. So we synthesized gadolinium complexes and attached them to the polymer and measured the T1 time for like as a dependence of the the gradual decomposition of the particle. The decomposition means we are basically liberating the gadolinium from the particle. In collaboration with Jörg Vrachtrup group in Stuttgart, we measured in the microfluidic channel and monitored like at the single particle level the changes in the T1. So here a little bit of chemistry, which is our love indeed. So we use three types of complexes. One was non-cleavable. This bond cannot be easily cleaved at any conditions we use. Basically, it's a diamond coated with a polymer. The polymer is shown here, right? And here is the non-cleavable, and these are two cleavable versions. Here is a cleavable bond, and here is another cleavable bond. They cleave in this fashion. So one of them is cleaved based on the hydrolysis by water, which is catalyzed by protons, by basically acid environment. So it's strongly dependent on the pH of the solution. Another type is cleaved by the presence of glutathione, which is present in the cells. Here is shortened as abbreviated as a GSH, glutathione, which is oxidized by this and basically is reducing this bond to, and again, liberating the gadolinium complex from the particle. So what we monitored was just the change of T1 of the particles with time. If we expose the particles to different environments. I will just show example very briefly. Now you see how it looks for pH 4.5 and pH 7.4. So here we have a little bit change. Maybe you can see if we fit it with the function, we get a little bit increase with a slight slope in the time, but it also almost constant. In this case, we have quite sharp slope. And note these measurements are within 1 minute. So that's our basically distinguishing time, because 1 minute is needed roughly for accumulation of enough data points to, to get reasonable error bar of the measurement. So what we will get if we make this T1 relaxometry at single particle level, so we get such a trace, that's basically change of the T1 time with the observation time, and it's fitted by the mono-exponential fit. If we do the same in a bulk and measure the number of the released gadolinium from the sample, we get this curve. If you have a look on of this time, like the constant, it's basically the same. So these processes are very well overlapping, and indeed they correlate together, because what we observe is actually the liberation of the gadolinium complexes from the particle measured optically by the means of the NV centers. So this sensor responds in the quite broad scale, which is physiologically relevant, from pH 3 to pH 7. And now we look how can we use it at the single particle level. So this is a bit more complicated. We have two types of errors. One type on this the triangle arrow. So this is short T1 time and long T1 time. What we did was quite actually simple. So we added these particles and let them to just to stay in the environment for time of about, I don't remember, a few tens of minutes. So basically they were, all the gadolinium was removed. And then we get the new population, like the new portion of the diamonds, and put them there. And we were able easily to distinguish them based on the T1 time and see which population came as a newcomer. So just showing you how you can average it for the different types of populations and distinguish them at a single particle level. If we detect it in the like this level, we were able to read out of about this like 10 to the power of minus 22 mole of conversions at one particle level, which is basically corresponding to few tens of or thousands molecules. So it telling us it's very sensitive, in other words. Still, we were interested to improve actually the sensitivity. What we did, we instead of such a polymer layer, which is shown like here, there was the polymer layer, which is quite complicated and also diffuse. I have to note that this layer is about 15 or 20 nanometer thick. So you can imagine we have a distribution of the interactions from the top of the level with the the level which is very close to the particle surface. So what we wanted is to kind of normalize the thickness of the overall layer and have all the interacting gadoliniums in the same distance. And this is why we developed this new like type of surface on diamond, which is basically covered with a very thin silica shell, which is supporting the formation of so-called supported lipid bilayers. This is the structure which is forming cell. It's a phospholipidic bilayer, but the main point is that the bilayer is actually really just bimolecular. The thickness is only 4 nanometer. So if we concentrate on the diamond, here is like idealized picture, but the point is that even if the diamond is like a different shape, it has multiple vertices or edges. Basically, the layer is going all over these things and putting all the molecules which we attach on the diamond level in the same proximity. We have actually two populations, one from one side, other from the other side. We can average them, and this is just showing what we did in the molecular level. We have such a molecule connected with gadolinium. We created this lipidic bilayer, and with collaboration again with the Jörg Brachter group, we modeled this and got a quite nice fit, which was actually describing the interaction, or let's say the change of T1 with the molar content of the gadolinium in the layer, in the phospholipidic bilayer. What we saw was nice such sigmoidal response within 0.01 the percent of the gadolinium to 1%. You can see it's actually order of magnitude more sensitive than the polymer, which is corresponding with the more defined thickness of the layer and the closer proximity of this layer to the diamond surface. So this is actually also the way how to improve the sensitivity and create something selective. We are now working on installing actually different molecules in this layer, but it's synthetically quite difficult actually to put everything together and get stable coated diamonds which contain exactly the geometry which you are expecting or want to have there. So yeah, that's our current research actually. So in our very recent work, we were following that polymer chemistry which we introduced like a couple of years ago, but now we pose a different task. Actually, can we use directly radicals for sensing? Not the gadolinium, which is not changing its spin, but if we can switch it to radicals. And we use like the so-called nitroxide, which are like shelf-stable radicals, which you can store in the lab at the room temperature. They are not degrading actually. So on the other hand, they can, like the most selective reaction, which is well known, is the reaction of the nitroxide with the ascorbate. It's a vitamin C, which is like present in many cells, especially in the plant cells, but also it can be used for treatment of humans. We actually is essential for our body as well. So we use this, and we were curious whether can we read this reaction dynamically using NV centers as a chemical process, which is occurring on the surface of the particle. So what do you get at the end? It is such a cyclic hydroxylamine, which is diamagnetic molecule. It's not interacting with the NV centers. So at the beginning, we have such a polymer-coated diamond with nitroxides. We provide this redox reaction, and at the end we have a, just this is actually in the middle of the reaction, and at the end we have just the cyclic hydroxylamine. So we can read out at a single particle level using the T1 relaxometry, as I was talking about it previously. So we see the change of the T1 with basically the progression of the reaction. A bit in more detail, the polymer extraction which we use here is a different one. This one was introduced by Komatsu, and we utilize it with some advantages for our the design of the particle, because we found this polymer can be grown in quite a thin, much thinner way than the HPMA which we used for the previous project. So this is now we have a polyglycerol on the diamond, and we sense this reaction as you saw. And if we have a little bit closer view on this diamond, this is the picture from cryo-TEM. Cryo means that you take a solution of the diamonds and you freeze it so quickly that the water, it doesn't crystallize. So it's not interfering with the process, and you basically have a kind of like the picture or shot from the nature state of the particle. In other words, if there is any polymer around the particle and you are able to see it, will basically you are observing its in natural conformation. So you can determine the volume of the polymer around the particles. So you can see this is like automated image analysis of this cryo-TEM, and we were quite nicely collaborating with the theoreticians which were using later the distribution of the shapes, sizes, volumes of the polymer and the diamonds to demonstrate, like to correlate actually the changes in T1 with the observed features, like the chemical processes. It correlated very well actually. So we were happy to see this. So we were measuring this using relaxometry, but also like in bulk, as we prepare like samples in a bulk. So we control all the processes using classical EPR to see what we see, what is going on actually with the radicals. So at single particle level, we were able, these are like different types of diamonds, ND1 to ND5. We have increasing number of the radical concentration in the polymer shell. So we use it for kind of like calibration of the T1 response to the radical concentration. And you can see quite nicely, we can measure it for like single particles and see this dependence. If we express it as a T1 for the many tens of particles which are averaged, it's depending on the increasing number of radicals per particle. You see these quite large errors bars. This is actually exactly what is the classical problem of diamond particle. This is kind of this advantage, because the particles are differently shaped, differently sized, and also they have like different, let's say, number of the spins which are naturally interacting with the NV centers. They also have different number of NV centers in each particles. So each of this quantity is distributed, which is broadening actually the effect, and this is making the preciseness more complicated or differing, like difficult. On the other hand, the sensitivity is kept indeed, but this is a kind of complication which we are now also thinking how to overcome. It's actually make all these parameters more narrow distribution of the like, let's say, sizes of diamonds, the shapes, the T1, everything makes narrower, which brings like the selectivity much higher, the sensitivity much higher, and is decreasing the size of this error bars. Okay, so what we did later, we observed as I showed you, and if we added the ascorbate to such particles, we nicely can observe like the classical exponential behavior, which can be attributed to chemical reaction. It actually can be used for the like determination of the ascorbic acid in the solution. We didn't demonstrate it in the paper, but it actually works. So this is how it looks before the reaction. There's an EPR spectrum where you see always diamond plus the nitroxide, which is completely disappearing, like at the final point. Actually, already at this point, within a few minutes, it's basically over. So yeah, that's kind of molecular readout. So here I wanted to show how to utilize, like, the, let's say, kind of smart chemistry, we can call it, as now it's commonly called, like the design chemistry for the transformation of the processes, which are even not necessarily paramagnetic. They can be read out using the NV centers by optical means with very high sensitivity. Again, notably, actually, the sensitivity of the processes are many orders of magnitude lower or higher in terms of, like, okay, sensitivity is actually higher than the other forms, like the fluorescent, for example, readout, because there are no means how to reach such a selectivity using a classical fluorescence readout. So NV centers are very sensitive sensors. You just need to somehow find out the way how to communicate with them and utilize them as sensors. So we and others are continuing now in these efforts just to improve it and bring it actually to cell. And that's the point, which is very important. I will just check my time. Oh, I think I'm quite yeah. So we started, like, Olga, when did we start? I cannot hear you. Sorry.
Peter, please continue. It's very interesting.
Okay, thank you. I want. Lecture. Yeah, I don't want to actually talk too long, so I will talk 10 more minutes. Okay. Okay, I try to be brief. So sorry for this. So okay, so the cells can be seen by different, fields of research in different views. You remember this picture? Actually, I took it from one review which is written by physicists. So this is how the cell is seen by physicists. I want to just show it's like pretty schematic, and it's like let's say constrained space where we want to determine something which is of our interest. So chemists sometimes see the cell as a chemical factory. There are thousands or actually tens of thousands chemical reactions which are occurring in the cells, and chemists are interested in a selective measurement of just one particle reaction or one particle parameter, and they want to find this out within many different things. Biologists see the cells also in a different way, as a biological machinery which is still like moving, developing, actually changing in the time. So there are very different views actually, and but I will show you that maybe some of the views of the cells, how did you see them, maybe not so accurate. So in the high school, you were told like the cells is terminated by a phospholipidic bilayer. He will see some cell, and below that there is a kind of cytoskeleton which is holding or keeping the cell in like the basically in a shape. And on the surface there are some receptors, and they can interact. They can be embedded in the membrane. Actually, each cell is coated by so-called glycocalyx. This is the barrier which the cells is using for protection against viruses, against bacteria, against many other factors which are negatively like influencing its life cycle and vitality. So this glycocalyx is around every cell, and it's actually very thick. It's thick, actually few tens of nanometers. So is the size actually of the diamonds which we use for the sensing, right? So this is one obstacle actually which we need to overcome, but not only this one. We need to get the diamond inside the cell and get it at a certain place. So how to engineer this? If you take a diamond and put it in the biological environment, it immediately precipitates. So this is caused by the creation of a mixture of adsorbed species on the diamond surface. It's actually occurring on any type of inorganic nanoparticle. It can be gold or titanium oxide or diamond or whatever. It can be even polymer. It always happens. because these layers interacting with the ions. For example, in your blood, there is 0.15 molar concentration of the sodium chloride. So indeed, these ions are interacting with the diamond. They are ions from the dissociation of water, like H plus or OH minus, proteins, DNA, and so on. Everything is interacting with the diamond, is absorbing on that, forming kind of, electric double layer, and so on, which is adversely influencing the behavior in the biological environment. So already with ionic strength for about 10 to the power of -4, the particles start to precipitate. Their size is increasing in the solution. It's also the same is valid for the pH. Within certain range, I mean the physiological range is here, at low concentration of diamond, they can survive. But if you go higher or lower, they start to precipitate. So you get something at the bottom of like cuvette like this. So also diamonds are interacting with the proteins. Each type of protein is interacting with the surface with different, kinetics, absorption, desorption rate, affinity constants. They are actually forming multiple layers even on the particles. So these are factors we need to overcome. If we use just, bare diamonds without any surface treatment, it will always happen, and basically it's blocking the surface from the purposes which we want to use them in the biological environment. So main contributions to this are like van der Waals effects, which work at very short distance, less than 1 nanometer actually. There are like electrical double layer effects, which are caused by the accumulation of these like the ions to the charged surfaces, and they are actually causing like the second layer, which is repulsing the particles. If you expose them into solutions of electrolytes, the particles start to get closer because these layers are contracting. They are contracting at the high ionic strength, and once you hit actually this critical distance, the particles start to interact very strongly, and basically there is no way how to dissociate them back from this status. So one way how to prevent this is use the polymers actually and develop so-called steric repulsion, steric protection of the particles. Taken all together, you can see these forces are actually very strong. These repulsion forces between the polymers, they are much stronger than these electrostatic forces. And if we take all this together, like here, we see overall effect, which is repulsive, and now is attractive thanks to the van der Waals forces. So we need to keep the particles really non-interacting in the aqueous environment with high ionic strength and even with proteins. So the polymers are a good solution. We can absorb the proteins electrostatically, covalently attach them. There are multiple ways how to cause it. We can also attach the polymers from the solution or grow them from the solution. I will not go now into detail because I want to fasten up my talk. So I will skip these slides. There are multiple polymers which can be used for this. We developed several of them and actually used them for protection of the diamond surface. What we reached was the very good protection in extreme actually ionic environments, and we introduced this so-called click chemistry, which can be used for attachment of different moieties. So this is kind of platform which we use for different studies, and what was the most important was to use it for the targeting of cells. So basically it's about taking the particle, get them into interaction with the cell membrane and get them inside the cell. That's the overall target, overall goal of this. So the problem is that if you take non-targeted particles without this, like the polymer equipment on the surface, and add some targeting group, something which is interacting with the ligands or glycocalyx on the cells, you observe some effect. That's right, but it's not very strong. It's maybe several fold increasing of the affinity, and that's it. That's what you can reach. On the other hand, if you will decrease the non-specific absorption, if you will basically make the diamond particle invisible. So it's not interacting with the cell. So then the specific effect is very strong. So the difference is you will basically gain the selectivity of interaction. And that's what we did in several studies. For example, here we attach this so-called GCP2 inhibitor. The GCP2 is a protein which is on the cell membrane, and we saw a very strong actually uptake. It was like 80 times more when the ligand was present there. So if you compare it to diamond, which is without the inhibitor, it's basically not binding. It's at the level of background. So that was from our point of view very strong achievement, because with other designs we reached maybe 5 times increase or 12 times, but this was 80 times. So we actually went even higher, and we attached transferrin to the diamond particles, but at the appropriate type of ligation and geometry. And what we observed was almost like 200 times increase compared to the selectivity. These are cells expressing actually the really high levels of the transferrin receptors. These other are not expressing such a high level. So you can sort them based on the interaction. If I magnify this axis now, you see actually that the levels of the interaction are so low in the just a few units of the fluorescence compared to about 200. So it's really showing we very well protected the non-specific interaction with the cells and boosted very highly the specific uptake by the cell. So finally, my last 2 slides. We utilize this for non-biological study, but it was again the same effect, interaction with the surface. So if you expose particles to some interface, they are always absorbing. This is quite a non-specific effect. And if you take diamonds and put them, for example, in combination with graphene, they will interact quite strongly. So you barely cannot dissociate them from them, wash them out easily. So we were interested in our collaborators in so-called dynamic covalent chemistry. It means we are basically utilizing very simple reaction between amine and aldehyde to form such a kind of shift base. It's called shift base, which is so-called a reversible covalent bond. It's a very strong bond. On the other hand, at certain conditions, it can also dissociate back to the original moieties. So there are only a few types of bonds providing this. And the goal was actually to take diamond, because it can be monitored very well without any degradation, and to put it on the graphene surface in a selective way. So we installed such kind of polymer on the diamond and equipped it with these type of aldehydes which can interact with the modified graphene which contain the amines. All of this chemistry was actually, it took us about 2 years to tune the chemistry, because there were many different types of moieties which were interacting too strongly. So it was not able to dissociate, or was just not binding well, and so. So, but there are groups which are binding very well, like these, which you see on this picture. And we created a pH gradient on the graphene. So the graphene was in the microfluidic cells, in cell. We created a pH gradient, and this was actually driving force which enabled dissociation before and before and behind the particle of these bonds. So the particles start to move, but the main point was it was not dissociating from the surface. It was still kept reversibly on the surface and moving, driving like a car, on the road. So we saw it for different, many hundred different situations. One of those you can see here. I hope I can start it. Okay, let me switch my because with this laser pointer it doesn't work. So I need to it looks like No. So
Мощности указателя. Мощности указателя.
yeah, I see. But if I switch this off, yeah, okay, it works. Okay, good. So you see in this in this rectangle, you will see a black point. This is diamond in a turf imaging mode from the confocal turf microscopy. So so if I start it, maybe you see as it is moving now. Here you see. So this is actually real-time movement within about a few microns on the pad. So it traveled quite quickly, you know, many hundred times of its own distance. So we observed many such trajectories and analyzed them later, you know, as the trajectories of the diamonds, and did some statistics and observed the dependence on the gradients and pHs. So for us, it was again proof that the proper chemical interface on the diamond can be used for very specific tasks and can be tuned for particular purposes. So at this actually point, I would like to end up my talk. Here are a few points which I want to just emphasize, that I was showing that diamonds are really sensitive as a sensors. Just what you need is to basically translate the language of the other molecules or the other quantities to these optical readouts, which is provided by NV centers. And if you want to use them in the biological environment, you need to design again the polymers for these purposes, combine it with different groups which are allowing the diamonds interact with cell surface, be taken up by the cells, or even be targeted inside the cells to the particular places. So this is kind of combination of physics and chemistry, macromolecular chemistry, and a bit of biology. Again, I would like to thank all my collaborators, which actually we are not able to work without them, because all these measurements and things are pretty complicated, and we are happy to work with them and with you. So thank you for attention, and I will be happy to answer your questions.